Here is a brief overview of the main Persephone functionality. The Persephone application runs in a web browser and can be launched by opening a URL. The genomes, structured as a tree, are shown on the left. We will start with a popular version of Arabidopsis thaliana genome. Selecting a node in the tree will open the list of maps, which are also shown graphically on the empty stage. To open a map, we will click here. This is the representation of a map with several tracks, some of which are hidden. The first one is the sequence track. At this resolution, it has the form of a GC content histogram. The poly-N regions are shown in blue, then the ruler and the track with gene models in two orientations, in the positive and the negative strands. The navigation is done by mouse. To zoom, roll the mouse wheel. To pan, just drag the map with the left mouse button, like that. To mark the region to zoom in, select it while holding Shift. Reset the scale by clicking the Auto Arrange button. Let's zoom into this region to see this particular gene. The tooltip will list the splice isoforms, which we can show in the track by expanding the strand, like that. A click on a gene model will open the Annotation Details form. It shows the properties of a gene, such as its name and location. The list of qualifiers is presented as key value pairs. Some of the values can be converted into hyperlinks pointing to external resources, such as arabidopsis.org in this case. The Metrics tab draws the exon structure of the gene model. The dark bar on the top of the gene glyph designates the coding region. A click inside the graphics shows various measurements. A double click will highlight an exon. This selection in the bottom part is synchronized with the sequence tabs on top. This way, you can see which part of the protein sequence is encoded by the highlighted exon. The selection is passed to the tab with the spliced sequence, the translation panel with nucleotide and protein sequences overlaid, and the unspliced sequence with color decoration that also includes introns. The Transcripts tab shows a summary of splice forms for the selected gene marked here with the bluish background. There are various options that help analyze the alternative splicing. Please note that the order of the transcripts here is based on the CDS length. If some forms share the same CDS, they can be collapsed to produce a smaller list. You probably noticed that a region of the genome was pre-selected. We call this a highlight. The highlights can be temporary, or you can save them for the future so that they are preserved between sessions. They can be edited, renamed, and assigned different colors. Let's take a look at a different type of track, which shows the RNA-seq coverage. This is an example of a quantitative track. We have two tracks with gene expression levels in different conditions. We can change the scale to the logarithm mode to reveal small peaks. To visually compare the expression levels, let's combine two charts into one by using the multi-trace mode. The differences in expression will be more prominent if we use the linear scaling. As another example of a quantitative track, let's take a chromosome from the human genome. This is also a quantitative track which covers every base. The values reflect the conservation level for 100 mammals. As you can see, we handle millions of positions here. It is quite clear that the conservation score is higher in the exons. Persephone provides the BLAST search interface. Let's take a quick look at how it works. We will take a protein product of a gene as a query and transfer it to the BLAST query by pushing this button. Just for simplicity, we will try to find the protein's original location in the genome. The interface allows selecting multiple genomes or limiting the search to individual maps. We will select chromosome 1 of Arabidopsis as the subject. Now we will run tBLASTN with the fast preset of parameters. Unsurprisingly, we found the matches on chromosome 1. The detailed alignment shows individual high-scoring pairs, with some of them spanning across introns and, as a result, having a low identity. This can be fixed by tweaking the BLAST parameters. Let's increase the gap penalties. The result produces much cleaner hits. The match can be visualized as a new track on the map. Persephone can also show the NGS read alignments, usually stored in BAM or CRAM files. Despite typically large data volumes, the histograms of the aligned reads for the entire chromosomes are shown within a second. Here we show a transcriptome alignment for one of the coffee genomes. At a higher zoom, we can see the individual reads. The RNA-seq data is a valuable resource for assessing the quality of gene prediction, measuring gene expression levels, or finding sequence differences. There are multiple display options for the alignment tracks. Some are turned on automatically, depending on the nature of the aligned reads. For example, if the reads contain methylation signals, the color schema changes to reflect the methylation scores. A click on the read opens a form that shows the read properties, such as base qualities or the read sequence itself. The majority of the Persephone data resides in the database and is visible to all users. There is another layer of data that users can add for themselves. The data types recognized by Persephone are listed in the import dialog. For example, the users can add genomic sequences in FASTA format. If the disk quota permits, you can upload entire genomes with multiple chromosomes. New annotation tracks can be added by reading from GFF3 files. The regular BLAST and text search functionality of Persephone will also apply to the newly added datasets. The new tracks can be uploaded using this import form or by drag-and-drop method. Let's take a BAM file with RNA-seq data for Arabidopsis thaliana. The program automatically recognizes the file type. If we choose the local session, the data will be processed in the browser, without any transfer to the server, and discarded later when the session closes. Now we need to attach the new track to an existing map set. After selecting the proper genome, we will see the summary of the maps read from the file matched to the maps available in the database. If needed, the matching can be done manually. In the last step, we need to give the track a proper name and color. Now we can see the newly added track on the selected map. Please note that when indexed files are accessed via a URL, only small portions of the data are transferred on demand, enabling rapid visualization of files even if they are hundreds of gigabytes in size. Besides the import function, Persephone provides a powerful way to export the loaded data. This can be applied to a map region, or to an entire genome. The export engine can be used to save both the genomic sequences and track data. Additionally, it offers tools for feature analysis, as the interface allows filtering and sorting of the data grid columns. Let's open the export dialog for a map of Coffea arabica. The tree on the right controls the visibility of the columns in the grid. One of the columns that we can add contains the protein sequences translated from the gene models. We can sort it by the text in the column and see if all of the sequences start with methionine. If they do not, we might want to save the list of such genes for future analysis of potential errors. To extract such genes, we can filter the column by applying a rule leaving only protein sequences that do not start with M. Now we can save the visible rows as a CSV file, or, as long as we have sequences to export, as a FASTA file. The Options dialog allows for customizing the FASTA headers. We will add the ID, the name, and the description of the genes. As another example of the export functionality, we will extract the genomic sequence of some interval on a map. This can be done by highlighting a region of interest and calling the export for the sequence track. You can now customize the FASTA header and save the result into a file or the clipboard. As long as we are talking about genomic sequences, let's take a look at another tool called DNA motif search. Besides searching for the motifs, this interface provides an extra functionality for visualizing the genomic sequences. At first, the nucleotide sequence of 2 million base pairs is loaded into the view, allowing for quick scrolling through the text. The cursor position and selection in the text window are synchronized with the highlight on the map's graphics. For example, once you start highlighting the interval on the map, the sequence view will scroll to the highlighted position and select the corresponding nucleotides. The DNA motif search itself is quite straightforward. Start typing the motif, and its locations in the text window will be highlighted using different backgrounds, depending on the strand orientation. To display the hits in the graphics, push the button Find All. Please note that the search can be limited to the selected map region, or, what is more exciting, to the entire map. For example, searching for the locations of the centromere protein B binding satellite DNA motif will help locate the centromere in the mammalian chromosomes. Persephone calculates quick statistics for genome assemblies in real time. To access the metric sets of a selected genome version, open its properties shown via the Context menu and switch to the Statistics tab. If the map set consists of chromosomes and scaffolds, the statistics at first will be shown for all maps combined. Use the drop-down box to limit the calculations to the subset of maps. Here, we can see the contiguity values, such as N50 or N90 for chromosomes only. The distribution charts are also automatically updated. In addition to genomic sequences, Persephone can analyze features within annotation tracks. Statistics pages are available for genes on individual maps or all genes in the genome. To access these, open the Basic Properties tab and locate the icon for Annotation Track Statistics. This form displays various metrics for gene model components, such as exon and intron length distributions, the number of exons per gene, and more. One particularly useful chart is the intron length distribution plot. If you plan to perform splice alignment of NGS reads, it's crucial to know the maximum allowed intron size. Reviewing the length distribution can help you identify and ignore outliers with extreme values. If needed, you can display a sample list of the genes in each range and study them individually. Persephone features a powerful search engine, accessible by selecting this item in the top menu. While the first tab allows you to search for a keyword across all categories, you'll get more accurate results by narrowing your search to a specific object type. For instance, if you're looking for markers with the keyword Alzheimer in their qualifiers, using the Marker tab and limiting the search to a specific map set will yield more relevant results with fewer false positives. Let's use the search interface to find map sets of type genetic. We will switch to the tab called Map Sets and limit the search to genetic maps. Then we will use a wildcard, an asterisk symbol, in the keyword field. The search result shows several genetic map sets. Let's open one of them. Genetic maps feature markers with locations in centimorgans and traditionally are shown vertically. The map orientation can be adjusted either for individual maps or applied to all maps. For now, we will choose the vertical orientation for all maps. The markers in the marker track have labels with their name, which can be clicked to show the marker properties. First, please note that each marker can have an associated sequence. This sequence can be used to map the marker onto the genomic sequence. Then, please take a look at the tab with all locations. This list shows all locations of the marker on different maps. We will use this control to display another map on the screen. The location field shows the mapping coordinates in base pairs, which means that this map is based on sequence. Now we can see the second map zoomed in to show just the selected marker location. Let's auto-arrange the maps to show them side by side. They are linked by the connector lines drawn between identical markers. Aligning genetic and physical maps helps reveal inconsistencies in assembly or indicates regions with a low propensity for crossover. The Persephone platform offers tools for data loading and processing on the back end. For instance, it allows the creation of artificial markers by extracting small sequences at regular intervals from a reference genome. These sequence tags can then be mapped onto other genomes, providing a fast and efficient method for linking and aligning maps of organisms with high sequence similarity. When identical markers are linked, the connecting lines indicate regions of synteny and structural variations. Here are chromosomes from two rice varieties, linked by common sequence tags. At this scale, we can detect large structural rearrangements. The connectors between the tags act as guidelines, pointing to the areas of interest for more detailed study. To place the connected tags against each other and bring the maps into the same scale, right-click a marker and call the Align Connected Features menu item. To study the alignment on the nucleotide level, we can compare the genomic sequences directly. At the scale of entire chromosomes, the sequence similarity can be found by engaging Minimap 2. This is done with a click of this button. When the maps are oriented vertically, the visible sequence on the right will be passed to Minimap 2 as a query and compared to the neighboring sequence on the left. Once the visible segment covers less than 1 megabase, the button for BLASTN becomes enabled. The amount of details in the BLAST hits can be overwhelming, so, at lower zoom, we should be ready to see just the HSP outlines. Once we zoom in deeper, the HSPs will be shown with all nucleotide mismatches and indels. The edges of small indels are drawn in red color so that you can distinguish them from the mismatches without zooming in to the single base level. You might wonder, why do we need the tags if we can simply compare the sequences? Well, to determine sequence similarity, we first need to identify which maps or regions should be compared. Many genomes consist of thousands of unordered scaffolds, making it unclear which ones can be aligned to a chosen reference. Take, for example, the region around the HLA-F gene on human chromosome 6. In our pangenome collection, we have hundreds of other human genomes, but their sequences aren't assembled into chromosomes. To figure out which scaffold in a specific individual's genome corresponds to the region we're studying, we check the All Locations tab for one of the tags. After finding the individual in the list, we click the eye icon to open the matching scaffold. We then adjust the sequences to the same scale and use BLASTN to compare them, making sequence differences easy to spot. To overview the synteny of entire genomes, we can use the Synteny matrix, a dot plot where each dot represents the link between marker locations on the maps of two genomes. The synteny is represented by diagonal clusters of dots. For closely related species, we expect a strong diagonal line with some gaps or inversions. Here, we compare maps of rice japonica and rice variety IR64. We can see the familiar inversion in chromosome 6. The inversion is actually more complex. To see it closely, we can zoom in the dot plot, select the cell, and display the two aligned maps. The secondary inversion is clearly seen. If necessary, we can invert one of the maps and untie the knot in the middle. You can also see a new type of track. Here, it is called inversions. It stores only regions that are inverted relative to other maps, but in general, this type of track is designed to show the synteny blocks. Clicking on a block will call the matching map on the screen and align the matching blocks. The synteny track can be used to show the order of scaffolds based on a reference map. We can see it in action on maps of the pomegranate genome. When dealing with organisms whose genomic sequences diverged over millions of years, using the DNA sequence similarity for the map alignment might not work well. In this case, linking the maps by orthologous gene pairs would be a much better approach. The orthologs are found by comparing protein sequences, so the search sensitivity is much higher. This picture shows the alignment of two chromosomes from rice and sorghum. Each line connects the orthologous genes. If we zoom closer and run BLASTN, we can see that the DNA sequence is conserved mostly in exons, and the identity of the matches is not high. A click on a linked gene will list its orthologs, which can also be brought into the view if needed. Linking maps through orthologs enables the construction of Synteny matrices for pairs of distantly related species. Here's how it appears for the rice and sorghum genomes. Persephone provides a powerful module for visualizing and analyzing genetic variation, including single nucleotide polymorphisms, insertions, and deletions. Let's briefly review this functionality. The variance interface can be called for a specific map. Here we can see the list of genotyping samples available for analysis. In this demo example, we have the data for 3,000 rice varieties, each with more than 4 million variant sites. The selection of the samples is done in the top panel. The sample qualifiers are available on the right. As in the other Persephone forms, this list of checkboxes controls the visibility of the columns in the grid. Let's display the country of origin. This column can now be used to filter or group the samples. To group rows by country, click the Group button. This view shows the size of each group and their average similarity. Clicking the Ungroup button will remove the grouping and return to the original view. One of the columns here is similarity to the reference genome. The numbers in this column range between 0 and 1. They will allow us to sort the samples based on the genetic distance. Let's select a few samples with different similarities. The selected samples are displayed in the central panel, linked to a histogram at the bottom that illustrates the distribution of variants along the chromosome. Alleles matching the reference are shown in blue, variants differing from the reference appear in red, and loci with heterozygous alleles are highlighted in green. Sorting the selected samples by similarity is instantly reflected in the graphical preview. The histogram can be changed by using a sliding window, which sometimes makes the picture easier to overview. The values in each column can be used to control the label colors for sample names. For instance, by clicking this radio button, we can assign distinct colors to labels for samples from different countries. Besides using the genomic sequence as a reference, we can nominate one of the varieties as the reference genotype. Right-click the sample and set it as Parent 1. Now all other samples are compared to the new reference. Similarly, we can switch to a coloring schema called TwoParents, for which we need to nominate the second parent. The preview can be zoomed in to a certain level, but to examine individual bases, we will open the map with the new track added and adjust the scale to the nucleotide level. If we're interested in allelic distribution around a particular gene, we can zoom in until individual variants are visible. At this scale, the local variants form can be accessed by clicking on one of the variants. The interface that opens will display genotypes in a grid format. The selected samples appear at the top of the table, though the grid includes all samples available for this genome. Each column header provides allele statistics. We can add a column with sample qualifiers and group the rows by its values, which can help identify irregularities in allelic distribution across different groups. Another use case might involve identifying which varieties have different translations for a given gene due to genetic variation. When the target gene is in view, a context menu option, Analyze Variants in Visible Region, can be called. Submitting this form will add a column to the grid with the sample properties, making it easier to locate the samples with translation changes in this gene. The formula for the column is defined here. The score calculation can consider only variants within the gene's CDS, or those that specifically alter its translation. The gene models should be read from one of the annotation tracks, which can be defined here. Now we can pick the samples with translation changes and study them in detail. The lower values of these similarity metrics will point to the varieties with stronger alteration of the protein sequence. When the mouse cursor is inside a CDS region, we can examine the SNP effects on translation. The red flags point to the variants that result in amino acid substitutions. The modified protein or DNA sequence can be copied from here. The local variants form shows alleles that cause the translation alterations using these little frames. To study a particular site, we can filter alleles, hiding those identical to the reference. Note that there are heterozygous variants where only one of the alleles changes translation. We can view this effect in the translation interface. When we add one of such samples to the track and open the translation form, we see this green flag that marks the heterozygous calls, where only one allele changes an amino acid. When we click on this DNA position, we can select which allele to use for generating the protein sequence. This is all we can show you in our brief introductory video, but Persephone is capable of much more. Please visit our website to view in-depth video tutorials and step-by-step user guides. Thank you.